Okay, welcome back. This is Issues in Biotechnology, the way we work with life, and we're in part 2 of the class on the applications of biotechnology. This section is on medical biotechnology, and this is comprising several different topics. We will start off with looking at a history of medical research and as it applies to especially biomedical devices, and then give an update on stem cell research, both embryonic stem cells, adult stem cells, and induced pluripotent stem cells. And I think that's a good lead-in into animal cloning, as some of the techniques are similar, and then how this could apply potentially to human cloning. And then in the second part of this series, we'll look at gene therapy, tissue engineering, and nanotechnology. The rest of medical biotechnology will also examine pandemic flu, both from a historic perspective and what we can expect in the future, and its conflict in terms of dual-use knowledge. We just saw the publication today in Science of the manipulation of the H1N1 avian flu virus and what that means for potential uses and applications both for good and for bad. And then lastly, we will look at cancer biology and emergent technologies in cancer treatment. So here we go. First, let's look at medical biotechnology from a historic perspective. But before we get to that, I think a backdrop to this is about infection itself, how infection has plagued humans since the very beginning. We have recognized in the first part of this course that self is okay, non-self is not okay, and that humans, along with many other organisms, have developed very sophisticated defense mechanisms against infection. There is an initial biological response, both by the pathogen and by the host, and then in humans, an immune response. And when we see in humans the development of antibiotics, antibiotics, since their inception, have been a tremendous boon to the medical world as well as the pharmaceutical world in this crossover. And you might want to think, how many of you would be here or would not be here had it not been for antibiotics? And today there is a lot of controversy over use and misuse of antibiotics, antibiotic-resistant bacteria, and I think a widespread misunderstanding about what they do and how they're used. So we don't really want to dwell on this part too much right now, except to say that there is a great deal of misunderstanding around about the use of antibiotics, not only in humans, but also in agriculture. We see the widespread use of antibiotics for the production of meat, and that would be beef, pork, and poultry. There are, in large agricultural situations, regimes where animals are kept on low levels of antibiotics throughout their entire lifetime. And this raises several controversies, not only in terms of food and food safety, but also the encouragement of antibiotic-resistant bacteria in the farm place. The situation is ripe for being able to select for those organisms that are capable of surviving in that environment. Some of those might actually be pathogenic on animals, but also on humans. And then there is the practice about how antibiotics are prescribed to humans and how they are applied by humans. Oftentimes, people feel sick, they go to their doctor and they ask for an antibiotic. not understanding whether it's a flu or a cold or perhaps a bacterial infection. Let's say it were a bacterial infection. Oftentimes, we will find patients who start to take the antibiotic regime, they start to feel better after a certain amount of time, and then don't run through the entire course of the prescription. Then the infection comes back, they then again dose themselves. And what this practice does is to encourage the survival of bacteria in the system that would be resistant to that antibiotic. And by repeated practice, we end up selecting for more resistant strains genetically, and these will eventually increase in the population. And this has occurred in a number of instances with certain antibiotics and with certain organisms. So there is a misunderstanding largely about antibiotics, their use, and also antibiotic resistance. But one thing we can see is that when any sort of treatment, such as antibiotics, or for that matter pesticides, or for that matter any anti-pathogenic treatment, we can encourage the selection of resistance. Only less than a decade after DDT was introduced, DDT-resistant mosquitoes were discovered. In fact, you can see this for almost any application. Agriculturally, we talked about the use of Roundup. Now the use and application of Roundup widespread in agriculture, we see the emergence of Roundup-resistant weeds. This is not at all surprising. In fact, you can probably find examples like this for any pesticide or antibiotic treatment. Take, for example, MRSA, multiple resistant Staphylococcus aureus, dangerous pathogen and resistant to, as the name implies, more than one antibiotic. Tuberculosis is on the rise. Why? Because it's resistant to certain antibiotics. In fact, there are strains that have resistance to more than one antibiotic. So what happens in a world when we start running out of the efficacy of penicillin? This is a problem. What are we going to replace it with? Certainly these drugs, predictably, have a shelf life. I don't mean a shelf life in terms of their storage, but their application. As we use penicillin more repeatedly, we will eventually encounter more penicillin-resistant organisms. That's just the way it works in biology. We've seen that already. So a selection pressure is applied, and you are selecting for mutants that are capable of surviving that pressure. That's the way it goes. So in the development of new antibiotics, we have to keep running and keep one step ahead, find new antibiotics, find new sources for new antibiotics, develop new drugs in this way to fight these organisms. Antibiotics do not cure viral-borne diseases. The flu, HIV, HPV, these are all viral diseases that are not typically affected by any antibiotics. And I have a friend who's an internist who repeatedly complains every year that I should state in this class that antibiotics treat bacterial infections, not the flu. For every patient that comes into the office and says, I have the flu, can you give me an antibiotic? Well, that's not going to help you. So there, I said it. We will be looking at the flu in the next couple lectures, namely the pandemic flus and historically how they've occurred, and look somewhat future into what we can expect next. The movie Contagion does a very good job, I think, of examining the reality of the next pandemic flu. Not the possibility of the next pandemic flu, but the reality. This is a certainty that there will be another pandemic. Historically, these occur cyclically, and it's really just a matter of time. So is this movie science fiction? It's not present day, but it might as well be a future example of almost a documentary. It's very accurate at the scientific level in its depiction, as well as its scientists. For once, we see Hollywood come out with a movie where the scientists are heroes, instead of vilified or misrepresented somehow. Could this really happen? Certainly, even to this magnitude based on what we've seen with previous pandemics. Should you be worried? A flu vaccine is not a bad idea. And keeping an eye on the news is also, I think, prudent. That said, bacteria abound. Bacteria are all around us all of the time, and they're within us. I don't know if you know this, but you are composed of 50 trillion cells. 50 trillion human cells, that is. About that. Maybe 49. But 50 trillion cells is about it. And you have 10 times more bacteria in you than you have human cells. So the microbiome, the ecology of bacteria that live within us and on us, is just now coming to light. And we just saw in the recent issue of the journal Science an entire review of current literature on the microbiome. Fascinating. The ecology of gut flora, how it influences our physiology and perhaps even our behavior. And the use of genomics to sequence those bacteria and evaluate how they respond to what we eat and our own physiology. It's fascinating. So in this class, I have been known to hand out Petri plates containing lauria broth, solidified lauria broth with agar, and every student gets their own 2 Petri plates and a supply of Germ-X soft wipes. Germ-X controls microbial hand contamination, or does it? So the hypothesis was stated, the use of Germ-X soft wipe towels diminishes the level of student-borne microbial hand contamination. And the experimental protocol that we use to examine this hypothesis is shown here, where 2 LB plates were labeled 1 and 2 with the student's name on them, or in this case, my own name. And the students were instructed to touch their 2 fingers from their right hand to plate number 1, and then thoroughly wipe their hands and those 2 fingers with Germ-X wipes, and then touch the same 2 fingers to plate number 2. Seal the plates, and I incubated them for 48 hours at room temperature and observed the colonies. So there's the exact experimental protocol here. And you can look at exactly how that was done. So in this experimental protocol, Germ-X towels, and this is not an advertisement per se, there are other products that do something similar, but the active ingredient that you can read on the label here is benzalkonium chloride at 0.13%. Inactive ingredients are water, polysorbate 20, glycerin, just things to wet the hands, phenoxyethanol. But this is the benzalkonium chloride is a broad spectrum antimicrobial that interacts with most bacteria and probably a broad spectrum of fungi as well. So the question is, first of all, how much bacteria are on your fingers to start with? That's interesting. Secondly, does this stuff work? So I categorized the plates from the student experiment into high contamination, medium, low, and 0. So by looking at the before, you can see on the panel to the left that big gobs of bacterial colonies are present in the ones that I categorized having high contamination levels. And that for the Petri plate labeled after Germ-X towel treatment, it's nearly completely eliminated or entirely eliminated. It's significantly knocked down. Look at the one on the panel to the right, midway down before. I mean, that's after 48 hours, that detects a pretty high and yucky level of bacterial contamination. I didn't key out the bacteria or do any taxonomy on these, but just to generally show bacterial contamination. And this would not be unusual. There are bacteria that live on our hands all the time. So this shows moderate levels of student hand contamination before and after. And I think you can see even from this slide, you can detect even the fingerprints and the imprint of the student's fingerprint with the bacterial contamination associated with it. And again, the Germ-X towel treatment completely eliminated that contamination. And low levels had some colonies associated with the before and either knocked down or completely eliminated after the treatment. And then there were some students who had 0 levels of hand contamination. I don't know if they either washed their hands beforehand or somehow confused the protocol and used the Germ-X towel before, but there were some, in fact, that had 0, and they also showed 0 contamination after. We did controls, which did not use Germ-X towels, showing that it was, in fact, the Germ-X treatment which eliminated the hand contamination on high, moderate, and low samples. Of course, 0 was 0 for both. And so the controls show that it was the Germ-X towels that had the effect on eliminating the contamination. I blocked out the student's name for privacy reasons. So the controls worked. The experimental results reflect the tested hypothesis. The controls, therefore, demonstrating a valid experimental protocol, shows that we have an adequate experiment in place to test the hypothesis whether Germ-X towels are capable of reducing student hand contamination. And there are the results in numbers, and they reflect what I showed you in the photographs. So the conclusion are that observed contamination levels on students vary. Fair enough, from high, moderate, low, and 0. There were different types of organisms that were observed, and they vary, including different types of fungi and bacteria with different growth rates. Some grew very quickly, some grew very slowly, as you might expect. And that what we see is that the Germ-X towels significantly reduced or eliminated contamination on all plates of plate number 2. I did this study with my little friend Nelson, in which I instructed him to first touch the plate, and we did the Germ-X towel and touched it again. He's 7 years old. And then, unlike the students, I had him wipe his hands with Germ-X and then put his fingers in his mouth and touch the plate. So that plate beforehand had no Germ-X, no bacteria, and then using his fingers as an inoculum, you could see that there was certainly bacteria associated with the oral cavity. And then his ears and his nose, and as you might expect. A human will touch their face as about 2,000 to 3,000 times a day. So all of that is transmitted to one's hands, and we all do a lot of handshaking. I'm not here to promote a fist pump, but perhaps after you look at this data, you might be encouraged to do that. So bacteria are all around us, so are viruses. And there are 10 times more bacteria than there are human cells. Some of them are our friends, and some of them are pathogens. So antibiotics pose right now an incredibly valuable defense against pathogenic bacteria in our environment. And we can expect that innovations of new antibiotics will come about from recombinant DNA technologies and synthetic biology. Now, instead of relying on fungi to produce antibiotics like penicillin, etc., that we could design molecules, as I described in the last lecture, that would uniquely fit sites on bacteria that would inhibit or stop their growth. And certainly we should encourage that type of research as the wolf is at the door, so to speak. Okay, imagine that you're walking across campus at night. You're alone. A person's walking towards you on the quad, and you feel understandably nervous. Suddenly the person grips their chest and falls to the sidewalk unconscious. You decide to A, stop and ask the person if they need help. B, that person is in cardiac arrest and administer CPR. C, keep walking. That person could be a mugger. D, turn and walk away. This is not my problem. E, dial 911 on my cell phone. So today, we have a cell phone in our pockets, many people. So unlike the dilemma on the train track that I posed during the pharmaceutical biotechnology lecture, this one has a technological solution that allows you to intervene in a way that was not possible previously. Before the age of cell phones, a surprising number of people would opt to walk away. And you can see by the response in my class that many people stop and ask if the person needs help, but the vast majority would use their cell phone. Why do we help strangers? This is a question of philosophical concern, but also to our topic today on medical technology. Actually, how often do people actually help strangers? This question has interesting evolutionary roots. By helping strangers, it's not immediately obvious what the advantages are to the individual. In fact, altruism itself is an interesting idea to study from an evolutionary perspective as well as a social perspective. As confusing as our understanding of altruism is, there is no doubt that it happens routinely in human life. GIs have been known to jump on grenades to save their comrades, not to mention firefighters, policemen, doctors, and in another strange expression, suicide bombers. It's also been shown in sociological studies that specific reasons why students, college students, would give for why they would help a person or not in need is that they would help a man or a woman who is more attractive. They would help a man or a woman who looks friendly. They won't help a man that looks mean. And they won't help a woman who looks unattractive. Now, this is interesting in terms of our perspective on altruism. It's kind of a sad comment, too. So, medical biotechnology. When do we intervene? How do we intervene? Why did we do this in the first place? What's the history that leads to some of the historical highlights of medical or biomedical engineering? We'll look at some of this history to show that humans have been intervening with trying to fix themselves for a very long time. And then we will look at how this applies to both cell and DNA-based technologies. In pharmaceutical biotechnology, we were primarily interested in the development of drugs. We can see now the development of so-called cellular pharmaceuticals, stem cells, for example, to treat diseases. A cell rather than a drug, a genetically modified cell. And then we will move on to animal cloning. So the techniques that have been developed for stem cell research have run in parallel with our ability to clone animals. Humans have not been cloned yet, but the technology, as I'll show you, is certainly in place. So if we look at a timeline of historical highlights for medical biotechnology, one that strikes me as interesting is the first prosthetic hand made out of iron with flexible finger joints occurred in 1504. So you could see that there was an obvious need, and this need was met probably by somebody who had enough money or prestige in order to try to fix that problem. Another interesting one is 1666, a blood transfusion between two dogs, setting up experiments that probably were prompted by need as well as curiosity. 1628, the repair of a human skull using a dog's skull bone. Well, I wonder how that went. Without antibiotics, without anesthetics, it is, however, notable to say that maybe this might be the first recorded example of xenotransplantation. That is, the use of another organism for human repair. Xeno-other transplantation. And we'll get more into this in the second part of the lecture on this series. 1863, the introduction of the first antiseptic surgical techniques. Now, this is pretty interesting because the United States is midway through their Civil War. So all of that carnage out there in the field eventually brings antiseptic surgical techniques. And we can only imagine the horrible situations at places like Gettysburg or battlefields where antiseptic surgical techniques were not conducted, let alone anesthetics. 1883, the development of Ringer's solution that kept organs alive outside the body. So 1883, already setting up a situation for organ transplantation. 1905, the discovery of growing human cells and tissues in culture. So this has been a long-standing developmental progression. 1928, the iron lung was developed to treat polio. Today, as I mentioned in a previous lecture, polio in the United States is nearly extinct due to the polio vaccine. But in those days, it was rampant. And so the development of an artificial iron lung to keep people alive was certainly needed. And it was need, as you can see in all of these examples, which bring about some innovation. Skipping ahead a little bit, 1956, the first successful bone marrow transplant. I'll bring this up later as a point of significance in stem cell research. Being able to transplant bone marrow as a treatment for leukemia was discovered first in 1956, 55 years ago. Owing all the way back in history to the beginning of the artificial hand, we see the totally artificial heart implanted in a human as a temporary measure in 1969. The first successful heart transplant using a human heart was conducted in 1967, and the patient survived for 18 days. You might think that's limited, but the patient did survive for 18 days. 1982, the implantation of the first Jarvik 7, a permanent, totally artificial heart. And then in 1984, baby Faye received a heart from a baboon, another example of xenotransplantation, and survived for 20 days, presumably succumbing to immunorejection. I'll bring this up later, again, when we talk about the use of genetic engineering of animals to overcome the issue of immunorejection for xenotransplantation. Like you might want to say, yuck, I don't want an organ from an animal. You might think differently if you were far down on the donor list and had fewer choices. So the Jarvik devices shown in this diagram of the Jarvik 7, or this illustration in 1986, was a totally artificial heart. Today, research has advanced, where in 2001 was the implantation of the ABioCore heart, which was a permanent, self-contained total heart replacement. Now it's interesting how the development of totally artificial hearts has progressed and actually plateaued. Many researchers today do not believe that artificial heart transplantation will go towards totally artificial devices, but rather a combination of muscle, heart tissue, and other ancillary devices that may facilitate that rather than a totally artificial heart. We don't know yet how future developments, what future developments in this area will bring, but you can think that recent computer advances should enable better devices, smaller devices, and so on. But these still have to be forthcoming. And there's a picture of the ABioCore self-contained heart in 2001. We've also seen development in other areas, such as the alloderm processed human dermis, which, as you might suspect, there is a large need for treatment of extreme burn victims. So whether it's artificial skin, skin grown in culture, there certainly is a large need for a product like this, and that has driven that. And recently we are seeing the advance of, instead of working on totally artificial organs, but engineering organs specifically, and then later I'll show you how tissue engineering is now developing. So here's an example of engineered human bladders. And there have been functional human bladders implanted into 7 young patients who have been recorded to survive, and they function quite well. Now we enter the era of genetic modification and impinging how DNA-based technologies and advanced cell technologies will become useful for the development of artificial organs as well as cell and tissue engineering. So in 1982, we saw genetically engineered insulin become commercially available for the first time. 1995, the first gene therapy trials. I'll talk about this later, but this refers to the ability to take a clone gene and the attempt to replace a defective gene in humans. You can think of examples all over the place where this might be useful. Coining the term genetic surgery. Say you had type 1 diabetes. Why not replace a defective insulin gene with a functioning one? Or most notably, other single gene genetic characteristics, SCID, so-called, severe combined immunodeficiencies, result in the so-called baby in the bubble, where someone has such a severe immune deficiency that they really can't be exposed to any pathogens. That's a single gene characteristic. And the focus of many early gene therapy trials. It's only been since 1999 that the first stem cells were first discovered to have pluripotency, the ability for a cell to become many other types of cells. And the first experiments with stem cells in 2003. More recently, 2007, the ability to genetically engineer cells so that they will behave like pluripotent stem cells. In this way, avoiding the controversies that have developed about embryonic stem cells and some of the deficiencies that have been noticed about adult stem cells. But more on this in a minute. The question is, that was posed during the agricultural biotechnology sector, and then again on the topics in pharmaceutical biotechnology is this natural? Is this natural, an artificial bladder? And why are some things okay and other things not okay? Can stem cells be used to create a new heart? If that's true, does the ends justify the means? Why are some things okay and other things not okay? Somebody brought up in a previous lecture that, the controversy about using genetic modification of plants for food raises a lot of eyebrows. However, using the same genetic modification techniques to make a protein that's useful for a drug barely raises any controversy at all. Same thing here, where some things are okay and other things not okay. But it really does beg the question, what is natural? We contradict ourselves all over the place, don't we? And sooner or later, this becomes even more significant. When do we start replacing human parts with bionic parts? Certainly, the advances of this have been tremendous over the last years, in part, sadly, spurred on by events in war. But artificial limbs have accelerated to the place where people with prostheses are competing in the Olympics this summer. I think that's fantastic. I saw a guy surfing the last time I was in Hawaii on 2 artificial limbs. This is great, and he was a lot better at it than I am. So when will we start to replace parts also driven by computers so that the longevity of these parts will increase human longevity? Is this a good thing? Certainly the need will progress the advances in these fields. And using computer parts now is at a forefront of this kind of technology. Not only material design, of course that's important, but using computer technologies are also interfacing with biology. And I will talk more about this at the end of this lecture in terms of nanotechnology. But certainly at the larger end of the scale, which I'm referring to now, computer-driven organs, certainly, but also organs that are computer devices that interface neurologically with our brain and perhaps our behavior. Experiments are already ongoing in this regard. But bionic restoration of hand mobility using nervous systems that will actually generate impulses to drive bionic prosthesis. So rather than simply a mechanical device like the 1504 flexible hand fingers, but actually having human thought generate an electrical impulse that then will drive that prosthesis. This is a great idea. And I think there are a lot of advances, and I think we will continue to see advances in this area. But computer interfaces, when does it get kind of wonky for us, Where there are people like Kevin Warwick earlier on here looking at computer chips to direct nerves in his arm to see if the computer could read and communicate signals directly from his nervous system. Where do we see this application? Well, certainly flying jets. Rather than relying on eye-hand coordination, which is limited, fast but limited, will mind control over these types of applications become significant. And you can see why this might be driven, right? But back on that, there have also been computer chips implanted in rats that could be directed by remote control through mazes. So by turning a joystick and saying right, left, the mouse or rat is directed to an eventual outcome. And you can, you don't have to put on your futuristic goggles too far to see how that would have applications in humans, and that might be a little scary, but we can also think in rodents that these things could go to places to find disaster victims, say from earthquakes, places where cameras can't go. So as we'll encounter in the lecture on pandemic flu, that we are entering a part of this course now where dual use knowledge becomes significant. There is a good and a bad side to many of these types of applications. Does that mean you just shouldn't do that? Well, I don't know if there's really much of a choice on that, really, but I'll get back to that later. There's also been advances on being able to send sound directly to the brain through cochlear implants. So hearing impaired people, I think, have taken advantage of some of these recent advances, and I think this will only continue to improve as the mechanics of cochlear implants and the technology improves. So, okay, we can replace parts. Where does the human begin and the part man stop? When do we become entirely mechanical with maybe only a mind left? Or when does the biology of the mind cease? Where now, is it going to be possible to download my mind to yours? I think it won't be as crude as sticking a USB port in here and linking it up, but that's not as far-fetched as this sounds. Or the ability then to carry nanoparticles that may have computing technology within us, and that this also could contribute to something that we think now of as the cloud. So an internetwork or hive mind of human society, so that the individual then becomes part of this larger organism, and that continuously updating and replacing parts begs the question really of human individuality and mortality.